Welcome to the MIP Discovery Series. In this video, I'm going to be joined by various members of the MIP Diagnostics team to answer some of the most frequently asked questions about molecularly imprinted polymers. First up, I'm joined by Dr. Antonio Guerrero, who is Director of Technology at MIP Diagnostics. So Antonio, we often get asked what type of targets can MIPs be generated to?
So, MIPs can be generated against a very wide range of target sizes. So they can target, for instance, very small molecules such as ions. For instance, MIPs are very good binders for phosphate ions. They can also be generated against small target molecules such as drugs, explosives, and toxins. That's also a very good class and size of target for a MIP. Next size up, we have peptides, so we can generate MIPs against a variety of peptides, or if we use a specific peptide that's on the surface of a protein as an epitope, we can generate MIPs against full proteins. Now, if those proteins are at the surface of a cell or at the surface of a viral particle, then we can generate a MIP that recognizes a full or a whole cell, or a full virus, or any fraction or segment of that virus. So in summary, we can generate MIPs for very small molecules such as an ion, like phosphate, or a very large full cell.
Excellent, thanks, that's a great answer. Thanks, Antonio.
Thank you.
Next, I'm joined by Alice Mazza, who is one of our senior scientists at MIP Diagnostics. So Alice, one of the questions we get asked quite a lot is how long does it take to develop a brand new MIP?
Right, new MIP, well, I'd say it depends on the complexity of the target, but typically the process is about 8 to 12 weeks. We start by molecular modelling to identify the best monomers to get a nice high affinity MIP. We then take the process into the lab where we produce and characterise the MIPs, and finally we carry out any refinements before getting the product to the customer.
Excellent, that's a lot quicker than traditional reagents such as antibodies.
Yeah, 8 to 12 weeks, I know, it's, it can be done.
Excellent,
for the right
great.
target.
Thanks very much, Alice. So I'm now joined by Shelley Bennett, who's a senior scientist at MIP Diagnostics. Hi Shelley.
Hello.
So a question that we often get asked is how do you actually choose the monomers that you use when you produce a new MIP?
So we have a specialist modelling software, and this allows us to screen the targets against lots of different monomers. The binding of the monomers to the target is then assessed in terms of hydrophobic interactions, hydrogen bonding and hydrophilic interactions. The best binders are then chosen and formulated into different compositions, and we test these in the lab.
Okay, excellent. And is there any sort of limitations? Can you analyse a full protein or a certain portion of the protein?
Yeah, so we analyse the protein and also sections of the protein, and we can then compare which section of the protein is best to target.
Okay, excellent. Thank you very much.
Okay.
So next up, I'm joined by the Head of Chemistry at MIP Diagnostics, Dr. Francesco Canferotta. Hi, Francesco.
Hello.
So with any new technology, we are bound to be asked how it compares with existing products on the market. So how do MIPs compare with antibodies?
Well, as demonstrated by one of our collaborators at Newcastle University, Dr. Marlos Peters, our COVID nanomips have demonstrated to outperform monoclonal antibodies. In particular, the sensor platform showed a greater sensitivity when nanomips were used as the binding agent, with achieving a lower limit of detection, about 20-fold.
That's excellent. And how about integrating them into a sensor device? Is that a simple process?
It is very rapid, and because we've got complete control over the surface chemistry, we can easily integrate these nanoparticles into many sensor platforms, depending on the chemistry that's needed for the coupling process.
Excellent. Thanks very much, Francesco.
No problem.
Okay, finally I'm joined by our Chief Scientific Officer at MIP Diagnostics, Alan Thompson. Hi Alan.
Hi.
So I've saved the most difficult question for last for you, and that is, what does the future hold for molecularly imprinted polymers?
I think the future for molecularly imprinted polymers and for us as a company is to get their acceptance in the marketplace. We've come a long way in doing that. We've taken the technology a long way from what people might remember of imprinted polymers in the past. And we're generating data regularly that exceeds anything achieved with imprinted polymers previously. So our job really is to gain that market acceptance and see MIPs in final products.
Excellent. And do you envisage any particular platform that will see steam ahead?
So far the adoption's been best with the next generation of sensors. If we take that to its logical conclusion, it's likely that we'll see MIPs enabling wearable sensors in particular over the next, let's say, 3 to 5 to 10 years.
Excellent. Okay, great. It looks very promising. Thank you very much, Alan.
Thank you.
Well, that's the end of our video series. I hope you found these videos informative, and if you have any further questions, please do get in touch with us at MIP Diagnostics. Thanks for watching.